Thank you, Silvana, for inviting me here and for this talk again on the same, meaning I usually talk about arbuscular mycorrhizal fungi, and I derive, I have a special interest on programming and computers, databases, apart from arbuscular mycorrhizal fungi. So I believe it will, because I have this unique combination of mycorrhizal symbiosis along with the computer. So I always enjoy sharing my experience and how things, are changing in our world, like when computer is just coming and programming and those algorithms are coming our way and things are changing. So thank you again for inviting me here. Do I start with my talk today?
Yeah. Yes, please.
Yeah, so I will share the screen now.
Perfect.
And yeah, so yeah, so I hope you can see my screen.
Yeah, we can.
So I will just remove this. Yeah, that goes fine. So we'll be talking about metagenome data analysis. And, I have taken the example of mycorrhizosphere, meaning how this data analysis methods and the techniques have been really, insightful in the study of mycorrhizosphere. So to begin with, I usually, very often I like to start with some funny note, maybe. So again, with a funny note, how this microbial profiling around, the two trees are talking that how the microbial profile around them is much like any other social media profile, is that in the soil we have thousands and millions of the friends networking together, positive, negative, stalking, anything, but usually we overlook them. And the metagenomic methods or ways, are giving us insight into these. And so maybe they get a better place than there's social media friends, or where, the interaction usually is overlooked. So I designed this talk with the objective, I just wanted to give you an insight how the metagenomic data, meaning collection of data and analysis of this data has been useful in interpreting, getting good results for and it becomes indispensable for the mycorrhizal studies at several locations. And then we'll also talk about how these studies have limitations, meaning where we are feeling limited because in these studies, and how the artificial intelligence or machine learning methods popping into these things and making them more useful for the present day scientists. And we would just have a small look at what are the future prospects of these type of studies. So we begin with my old paper in 1994, where I was studying mycorrhizosphere and mycorrhiza, and then I used to do it how? I used to do this with culture methods, and I collected all root exudate and collected the fungi around this root. And then I could establish a correlation between the exudation, the change in exudation because of mycorrhization of these roots, and the flora around these roots. But now we have different method over the period of time. I realize now, and this is my paper in 2017, I suppose, where I have thoroughly discussed, how the paradigm has shifted from culture studies in the rhizosphere to the microbiome studies, and how it is becoming very useful and has helped us in solving several of the questions associated and in the rhizosphere, and especially in the mycorrhizosphere. Because not everybody here is of the AM fungal background, because I see there are different symbiotic associations. So I thought it makes sense to first tell you what are these AM fungal microorganisms or how they associate before we talk about them in little more details. Is the mycorrhizal AM fungal symbiosis is the symbiotic interaction, as we know, is between the the fungi belonging to the phylum Glomeromycota. And from outside, we cannot distinguish whether this root is mycorrhizal or not because they don't have any outside symptoms. But inside, they produce structures inside the root, and in the soil, they produce spores. In the root, they produce typical arbuscules. And studying them is not easy. Again, this picture I have taken from my other paper. Studying them have never been easy. Why? Because we cannot culture them easily. And their taxonomy is very, very difficult. And earlier, when we used to identify them, we used to have culture-based method, meaning we used to raise pot cultures from one spore and then develop this to several generations because these spores usually are associated with the other soil microflora, and meaning it was a difficult task. Even now, it is not that easy because they can be cultured. Very few species have actually been cultured. Then we gradually shifted to metagenomic studies because when you want to take the environment along with the meaning taxonomy is one thing, but when you are studying the environment and the interactions, and you got to shift to metagenomic methods for such studies. And in metagenomics, it was all sequence and all sequence, and we left the biota part of this, and we used to talk about the sequences and the environment and the metagenome. And everybody appreciates these three terms. They became very frequent after that. And in this metagenome data analysis, when we'll be talking, we'll be again using these terms very routinely, like what is OTU is operational taxonomic unit, which is a proxy for the morphological species, which we used to describe earlier. So I would say OTU for any species, which I'm talking in the rhizosphere. And this term OTU is now gradually being replaced by ASVs, that is amplicon sequence variant. And for the sake of accuracy, because the cutoff in OTUs is 97%, and because of that, I maybe in few later slides, I'll be able to show that how that was leading to underestimation of species, and it was not giving the correct estimation. And basically in metagenomic data analysis, what we'll do is we'll isolate all the sequences from a given rhizosphere, from the given rhizosphere of a mycorrhizal root that we call as mycorrhizosphere. So once we have isolated them, amplified them, we'll go for their taxonomic assignment, and we try to give the OTUs which we isolated a name. And here lies the main important thing, which I want to convey to the scientists around also, is that what is the most important segment in giving the taxonomic assignment to these sequences, and how should we collaborate and work further to get best out of it. So I like to explain the difference between conventional genomics and metagenomic studies. Like when we used to do it earlier, say for mycorrhizal fungi, we used to grow them in pot cultures and then isolate the DNA from one spore and then match the sequence and then by simple genomic methods and give it a name. But this could be done only for maybe 1% of the species. We could not all species can be cultivated. And that was a very cumbersome process. And also, to carry this in the given environment was also hard. So this gradually, for environmental studies got replaced by the metagenomic method, where in principle, we could study most of the interacting organisms in the rhizosphere, and we could access at least most of the species associated in that region. So they are different from the conventional genomic methods where we used to culture, but this culture could be avoided in these metagenomic studies. And for AM fungal, especially the AM fungal species or AM fungal studies, we usually do not, I usually practice this amplicon sequencing based on the rRNA sequences. And usually, the 16S RNA sequence have been used. They've been ideally used for this purpose because in most of the, like for other fungi, we use ITS, but here we use 16S rRNA sequences because ITS doesn't give you the correct result. And these sequences have the additional advantage of being highly conserved and having both the conserved and variable region. And the biggest advantage is that we have the huge reference databases, because all these studies, they depend on the reference databases. We must have already identified reference databases on the basis of which we could say that this is this fungi or this is this bacterium. So we will again be focusing on the importance of reference database. So I was just telling you that the metagenomic data for fungi or for the otherwise, we could study them as markers, gene sequencing, or maybe the whole genome. Whole genome sequencing, but the whole genome sequencing is very expensive and it becomes a huge project, though it will give you a lot of information, like in the marker gene, where we are using identifying one marker gene and amplifying that and picking the OTUs and giving it taxonomic. We could know what all species are present, but in whole genome, once we have developed a draft genome, we could do a lot more studies and it gives you a lot more information. But here we'll be mainly focusing on metagenome data with the marker gene, and we have mainly used CHIME for this work. So I will gradually introduce because what is happening, but we'll be mainly doing the marker gene sequencing. So the image of the rhizosphere, gradually, when we were culturing, we could not produce this much of the data and this type of data that we can estimate at every region that the different distribution in the pie chart, how many bacteria, which bacteria, their quantitative, qualitative estimation, and a correct estimation which relates with the environmental parameters. This could be done with metagenomic methods, and this made the results really interesting from that perspective. And it gave another concept that we call as superorganism or holobiome. Like in my mycorrhizosphere, I could introduce several new concepts. Like when I isolated all these species, this picture I have taken from my paper, and the earlier one also was from one of my papers. Anyway, so this picture where I'm just drafting and telling you how the rhizosphere microbiome can lead to several important conclusions, and when we analyze these species thoroughly. So when we study the community, all community in the rhizosphere is we could identify what is the minimal community. What does it mean? Minimal community means, like I said, I have kept 1 or 2 here for this one. If I remove these 1 and 2, the plant will not be able to survive. So when we study all community along rhizosphere, we know that few of these species, they are must for the existence of this plant. This will not be able in the natural environment, it will not be able to grow if these 2 species are removed. These we call as minimal community. And then we have core community members. What are these core community members doing? If you remove them, the functioning will go, though the plant will survive, but the functioning of that plant will go in the given habitat. So the core community is necessary for the normal functioning of that plant in that environment. So like in here, I would say 1, 2, 3, 4 species, they are important members of core community. If we want this plant to function normally in a given environment, we must have these 4 species at least present in the right combination. There can be few other ways, this opened several dimensions of the studies and new studies. Like, there were certain species which were reported like, say, 5 to 8 here. If I remove these species, still the plant can survive and function normally. These we call as accessory microbiomes, meaning they are additional species. If I remove them or replace them with any other species, still the plant will be able to function normally. And this was what was the analysis of natural microbiome around the root, is that if we have 8 species of these, 4 are necessary for its normal function, but 4 we could afford to replace with the other species. And this was, gave to the concept of synthetic microbiome, in where what happens is we could replace these accessory microbiome species with, so yeah, with the other beneficial microbes and make it a better better for the system synthetic microbiome. The better maybe in whatever is your interest, if you want to make it disease resistant or if you want to make it better productivity and that way. So this, to us, this opened a new arena and a new field of study which we call as rhizosphere engineering. And what we do here is that we study the rhizosphere, we analyze all the genera or all the species, taxon present around, and what we do is we relate them with the several functions of this plant, and we could know that which are good ones, which are bad ones, and then we could replace them with some of these in the synthetic community, so that to get best out of this. And all these have been possible just because we could, with the metagenomic methods, we could analyze them both quantitatively and qualitatively, and we could study their interaction among each other. So the other thing, especially these studies were suitable for these symbiotic studies. Why? Because we have millions of the organisms in the natural environment, and they vary in sizes, they may be different in locations, and especially in our case, they were very suitable. Why? Because they are obligate symbiont, they do not grow without roots. And when we culture them, the infection is a very limiting factor, and we have to grow them for several generations to get good species to isolate their DNA, good spores or good material to isolate their DNA. And still the interaction, because that also is not possible for many species. So their interaction studies were really suffering in that. So when we shifted to metagenomic methods, it really made sense and opened windows of several new dimensions in this study. And another things with these symbionts is that if we culture them together or separately, the result very often varies. And even if I said already the culturing methods are known, they are very lengthy and tedious. So we have high throughput sequencing technology, and that allowed us to study, but still we needed to have the bioinformatic algorithms and methods developed to get them going and to reach to the stage where we can actually manipulate them and use them for better functioning. So this was just, I was trying how the scenario has been changed for rhizosphere microorganisms with these methods. And now I'll take you where, how do we study them? Meaning, how do we analyze this metagenomic data and how do we collect this data, and what is lacking to take it to the best or make it a really science which gives us the information, especially about these AM fungi. So how do we study the AM fungal community structure in the rhizosphere? I would say we have 5 main steps here. The first step is sample preparation and DNA isolation. Next would be DNA amplification. Then we check the whatever sequences we get, we do the quality check with them. And after the quality check, the important assignment is picking up the OTUs, meaning how many species or OTUs were there in your sample. And finally, we give them a name, like to which species this sequence belongs. I just I divided them into 5 steps, but it's not that easy and that simple. There are several, important things which needed to be taken care at every step. And I'll be showing you how we have done this with the help of the software, that is QIIME. And we have not used several other softwares which I have listed here. So we have several options at every step. We have several reference databases. We have different methods for OTU picking. We have different analysis software. What I say, I'll be using QIIME here for the sequences, for the analysis, metagenomic data, identifying OTU, meaning all these steps would be done with one software, that is QIIME. And we could use different reference databases. I'll tell you what all they contain, but we mainly prefer Marza OPIC for AM fungi. So these are the 5 steps in their analysis. So let's take them one by one. Is that what we do and how we do is, the first step is the sample preparation. And how we prepare the mycorrhizosphere sample is that we wash the roots in the water, and then with simple soil DNA isolation kits, they are available. We could isolate the DNA from there, and it's a routine protocol. But we must keep three controls here. One is a negative control, where the soil DNA is absent. The other one is a positive control, where our known species, meaning few of the species which we know they are going to occur there, we keep them as a positive control. And there is another type, because it involves vast number of species, and it is liable to error, and a small change in the sequence is going to give you change in the name of the taxon. So checking the errors and maintaining the best error-free information is, we run a third type of control here that we call as mock community. And what we do in mock community is that we have already designed expected set of microorganisms, and we make a artificial community, and we also run that along with this negative, positive, and mock community to get best and accurate information. So the other thing which we must know about our sample is that we have to have the primers. Like for our fungi, we use SSU, LSU, or ITS primers, but mostly it is because the database is available only for SSU. So we have to go for SSU. But technically speaking, for fungi, we should have used ITS, because the tree of life is built based on ITS. And for the other fungal identification also, we use ITS. But ITS for AM fungi does not give accurate results. So we had to shift to SSU sequences, and for amplification, we use next generation Illumina sequencing. And I have this small drawing which I have drawn, and I was just trying to explain you here how a small amplification error could lead to wrong information. Like in this hypothetical example, where we are studying the diversity and abundance of sequences from metagenomic data. And here each dot represents one organism, and one color represents one OTU or one species, right? So when we clustered them, it gave us 4 different colors clusters, but these 3 were confused, meaning they didn't know where to go, because they were not exactly like these, but they were similar to these. So with pairwise alignment, we could align them and keep them in group. The error ones lied along the side, but they did not qualify for becoming a new species. So by clustering, we could, and we just removed all the species which were less than 97% similar. So this gave us what result? This gave us the result that it has 3 taxons, because the green one merged with these and it took it here. So when we have a fixed cutoff for clustering, it leads to wrong results very often. And what I'm trying to tell you here is that if we have sequencing errors, that could misguide our result. But this OTU concept, as I was telling you previously, has been replaced now with ASV. ASV is amplicon sequence variant, where we don't keep we sometimes, we cannot avoid these amplification errors. So the amplification errors are still there. And with the pairwise comparison, we had these again giving the same thing. But when we, instead of, keeping it fixed, we allowed a lot of reiteration for the system to decide what should be the cutoff, meaning how by its experience the system is feeling by the learning methods, that what should be the cutoff. And when it was allowed to decide, it corrected and it gave us 4 species here. So these methods, these computational methods have really added to the accuracy of the result. And we have to work on the sequencing errors, amplification errors. But this, this amplification and primer selection for AM fungi, we worked with rRNA, with SSU sequences, and these we had to have stringent quality check about with these sequences so that these sequencing errors or these clustering errors does not give us false information. So now what information we got and what information we get after Illumina sequencing. We get 4 types of files. When in Illumina sequencing, it's more of the job of the robot. He will do everything for you once you give them your sample. And it gives you the result in the form of 4 files. One is a mapping file. The other one is a barcode file. Then we have a sequence file and a reference data file. So it gives you 4 types of files based on your input. And let's know what does these 4 files contain. Then only you will be able to do metagenomic data analysis, because you should understand what these files are containing. So one is the mapping file, which you actually supplied yourself for amplification. And this mapping file contained all the information about your experiment. Like you have in the one sample, you have maybe glomus rhizosphere, and in the other one is non-AMF or maybe colonized by the other AM fungi. So in the mapping file, you provide all the map of your experiment, the hypothesis which you are studying, and this is used all through the analysis because you provide your hypothesis here. Like I've just picked up one mapping file wherein it gives you the hypothesis. It tells you it's data from 2 individuals and it is taken from 5 body sites at different points. So the mapping file, if you open up the mapping file, it will give you a picture like this, and it will tell you the hypothesis of the experiment, that what actually you were working on. Then you also get a barcode file, because the person who has done this Illumina sequencing for you or next generation sequencing, he would give you this barcode file. What does this barcode file contains? It contains the barcode sequences and all the other information. And why these barcode sequences or this file is needed is because while running the Illumina sequencing, they sometimes mix all the samples together. And when they mix all the samples together, they call this as multiplexing. So when your all samples are together, before you start your work, you must have the barcode file so that you can assign each sample to which sequence belongs to which sample. So like when we go further in the few next slides, you would see how we could assign the different data to different barcode files to different samples. And this process of assigning sample to different sequence is called as demultiplexing. So before going for this metagenome data analysis, we must know whether this multiplexing has been done in your sample or not. If your amplification, your sequencing has only used one sample, then it is fine. But if multiplexing has been done, then you must have to do the demultiplexing. And the third file which you get is the sequence file, which contains your sequences. And these sequences are in FASTQ format. And FASTQ is a very varied version of FASTA format. And it gives you more information about the quality, because here in this study, the quality of sequence is very, very important. I just gave you one example some time back. If the quality is not right, it is going to give you wrong result. And the quality filtering, if you see the steps in data analysis, when we reach there just now, you will see that how important it is to have the quality filter. And the fourth file which you get is the reference data file, which is very, very important for our fungi, is because we have to have the correct reference data file. Meaning this contains the already identified sequences. Like it is the work of it is the work of extensive labor where the already studied sequence, they've been cultured and isolated from that organism and stored as a database. And these reference database sequences have to be very reliable and accurately identify. For bacteria, this has been worked, and we can get reasonably good identification for bacterial species, but not for our fungi. For our fungi, still to get accurate identification is a challenge. And we are in the process of building the databases. We have one database, but still, when we identify our species, we get more than 40–50% unidentified because there is no corresponding sequence available even in the reference data file. So when we get the result from Illumina sequencing, for our soil sample, we get these four types of files. And now I think I could explain what all these 4 files contain. And we must have all these 4 files if we want to go further with the data analysis. So, well, I was telling you about all this, and then I was telling you again that the quality of the sequences is very, very important. And I also explained you the barcodes, why they are important, because they are given, each sample is given a unique identifier that we call as barcode, and we have to have these barcode sequences, and we must get rid of them before we go for analysis, because any wrong base from there is going to give, wrong result. So we must get rid of these barcodes, primer variants, whichever has come in your result, and all the amplification errors. So the quality check, I would say, is the most important step in here when you're going for data analysis. And I thought I could demonstrate you this with QIIME 2, how to do the quality check, because maybe I have some results, some data, and if not hands-on, at least you could have a look how it is done. So the QIIME 2, why did we select QIIME 2? I find it very easy, that is how, and it's quite accurate, quite popular. If you Google QIIME, or maybe if you want to study the small subunit RNA analysis, metagenome, you would say you will get 2 softwares. One is QIIME and the other one is Mothul. Both are equally good. Both have their advantages, disadvantages, but we chose QIIME here because it is good for many things, because for every step they have a provision. So it was a complete package for us, and it worked well with our sequences. And this is open access. This has a very good troubleshooting community available, and it worked well with it was an open source software. So, but before we begin, we must have had this in our system, and this doesn't work with Windows. Windows compatible is not available. So we had to install Ubuntu before we begin, and Ubuntu or the Linux operating system. And again, this was very, very simple, because if you see nowadays in Microsoft Store, you get that as an application. Like you download any application, you could download this Ubuntu, this version, it works well with the QIIME 2. So from Microsoft Store, you could download this Ubuntu, and then I said it is not GUI, then with several steps and interact, but it is much like any software which you install in your system, and it's not GUI, so you get a result like this. I've just taken a snapshot that after installing Ubuntu, then you get a QIIME 2 written here. This shows that your system has QIIME 2 installed, right? So after done with this, once the QIIME 2 is ready, you must have all these files which we were talking. You have to bring this in here for their analysis. So you have to get all the files which we were talking, and we have again commands. I can tell you the commands here, or maybe I can provide them in a notepad later on. But in the quality control, we have 3 main things which we do here. One is demultiplexing. I explained you that already. And here we link the sequence to a sample. And the other one is quality filtering, is that we have a set criteria that if the sequence does not meet these quality parameters, that needs to be removed. And we have several softwares for doing this, but we do this with QIIME, and I can demonstrate how did we do that with QIIME. So in the quality filtering, we provide stringent criteria for the sequences to be selected, which are of good quality. There's another thing that is decontamination, because generally in soil samples, what is happening is the bacteria usually outnumber the fungal or the fungal community. So usually, so we have to do a bit of decontamination so that, the our species are not masked. So these three steps are there in quality control. And like this could be as simple as running any command. I just took this video in this PPT. I was just trying to show how this works. This looks very simple, and this is my laptop. So maybe you could install, you already have installed Ubuntu, and then you add chime to this, activate your chime, and then maybe you can, it is just taking, it will show you all your slides, and they are very simple commands. I can give you the commands. I'll be able to supply if anybody wants. So it has activated chime, and like for demarks, I said demultiplication, you don't have to do much. You rightly give your file names, right? And run this command, and it gives you the result. It gives you the result in the form of several files again. Like in here, if the result, if you want to see the result, it gives you the result like this. It, by the demultiplication, demultiplexing, it assigned your 91,785 sequences to 34 samples. So earlier it was 1, and by demultiplication, your one command, it assigned all these sequences, like if you can see, it gave you the result to the different samples, and it also gave you one quality score, meaning each sequence is having what quality. And this quality score usually is done on the basis of maybe minimum per read length, maximum bad run length, sequence. So we have several scores, and on the basis of that, the quality score also is given. So in this step, we run that command. After you have installed chime, you call that command as I showed you, and then you can assign your sequences to the samples, and you get a quality score also, whether you're going to get, meaning what is the good number of good quality sequences. And then the next step I told you is quality filter. Again, I have written the command here. You could pass this command and it will give you what, but mind you, here you have to write the names of your files, whatever name you give to your files. It gives you output. This O is for output, and it will give you the output for the filtered sequences, because it will leave all poor quality sequence from your data. Now you will just have your good quality sequences after this. And this denoising is another computational method which we must run, because it is a computational method, standardized protocols, because of which we are able to remove the sequencing errors, whatever best possible, so that when you're talking about 91,700, the number of sequences I told you just now, and that was the part of the experiment, that was not the full experiment. And so it will remove all the sequences which are expected to have an error, and it is already defined computational algorithms which are doing this. So after having done this quality filter, what we do is we pass the same sequences for denoising and OTU picking. And for doing this, we use the software Debbler. That's a part of chime, because chime is a package. We have this Debbler in there. So when you pass, the output will be what? The O is the output, and in the output you see what? You get the table, and you get the OTU table. So it would have picked all the OTUs, meaning how do they pick these OTUs is that they cluster them in the groups. So like this process takes time, because they pick up the OTUs, the clustering which I showed you in the previous diagram where we were talking of OTUs and ASVs. So they take all these 91,000 sequences and they cluster them in different groups. Each cluster is called as one OTU. And then they prepare what? The OTU table, meaning which cluster, how many sequences. And you get a table like this, a graph like this, which makes the table summary that you got these many OTUs in your sequences, it will remove all the sequences which does not cluster properly. I hope you remember the diagram which I had drawn earlier. And now it will assign one cluster to one OTU, and by the, maybe I told you that it can use the user defined similarity threshold, or it may be by machine learning, it will leave it to the system. By its experience, it can define what is the threshold to be kept. But we get a good OTU table like this. And the OTU table or the feature table which we have got, it tells us that we have, this is the each sequence, and this is the frequency with which it is occurring, in the number of samples where it is occurring. And with this information, we could identify this sequence by simple blast. If we run that in NCBI, it will tell you the name of this sequence. And the other method of identifying this sequence is by taxonomy, by the reference data, which I told you just now, is that you have to pass your already identified reference data, so that by similarity searching, your sequences are given a name, like I have just kept it here, which bacteria, which fungi, and it gives you a summary, like how many, meaning what about the abundance, quantity, and you can then make all ecological calculations with this. But the taxonomic assignment is the most interesting one, whether you get your result. And I said you can do this with BLAST, or you could also do the phylogeny, maybe phylogenetic placement by different softwares. And we have several machine learning protocols also, machine learning algorithms available here, like NB or RDP classifiers, you could use them. And we have several taxonomic databases already available, and more for bacteria. For bacteria, we could do good identification, but not for many fungi. And because what happens is, like, especially for our fungi, more than 50%, they will come as unidentified, because though it identifies that it is a OTU, but we don't have an already identified sequence for that. So these were just the examples of several databases which we have. Like the CHIME uses the RDP green genes, maybe Motor uses Silva. You could choose any one of these, like we use Marza AM for identification of AM fungi. Then again, we have several limitations when we are identifying sequences, AM fungi in the metagenomic data, because we have the collection based on SSU sequences, and the ITS sequences are used for other fungi, but we don't have a collection for ITS sequences for AM fungi. Then we really cannot compare many fungi together. And we get a very poor matching. I told you that, meaning more than 50-60% species, they remain unmatched. And it requires a very specialized knowledge and a very specialized sequence collection, which in case of AM fungi, we have very few experts who could tell you the right sequences or the right data, because in the data, again in NCBI or wherever you go, you see that the data quality is not very good and not the correct sequences are lying. And the other problem, which, because I had a paper, maybe I can show you here also, where we observed that if we go by this way, we will miss lots of novel sequences, new sequences which are coming, lots of rare sequences which are not there in the database and they will be missing, and lots of cryptic species which are not coming here. And we have to generate the training data, and the data should be of good quality. So when we are talking at an audience where they come from varied, they come from different backgrounds, different type of symbiosis. See, the point to be focused here is in these type of studies, the data generation, meaning the development of training data. Training data meaning where we train our machine to identify, and the database should be with the good quality data, so that we don't lead to wrong conclusions, because this type of study is quite dependent on the databases and the quality of database. So just a brief, I will just take 1 or 2 slides for this one, is that what is artificial intelligence and what is it doing here is that when we simulate the human intelligence in the machines, and with the help of programs, the computer start behaving as if it can mimic the actions. We call this as artificial intelligence. There are several types of this, but still, I think I want to skip this here. But this I want to show you here is that most of the metagenomic data analysis algorithms, they apply this machine learning algorithms, meaning at every step from clustering. I told you the clustering, how clustering is important in classification, in taxonomic assignment, in preparing the draft metagenome. At every step, these machine learning methods are used. So these, without these artificial intelligence, in fact, this conclusion I drew from my paper, which came just last year. And what we observed is that again, I have just summarized that. But the thing, which important point, which I was saying, that at every step, we are using the machine learning algorithms. And we need to work more on that. With bacterial data, bacterial taxa, we have reached to some level. We could get good identification of most of these sequences, but not with our fungi. For our fungi, we need to have more of good quality databases, already labeled data. And the other thing is the message which I'm trying to give you here is the communication among the scientists of different disciplines to solve this problem is that from their experiments, they should generate good quality data that can be stored and that can be useful for the other people who are delivering the algorithms, so that for further studies, these studies can lead to important ecological conclusions. So this was just, I was drawing for some other work of mine, the evolution of metagenomic studies, and I was just trying to add this machine learning methods are really have revolutionized these methods, and they have really come up as a boon for this study. Here are some references if you want to study this more, and this one happens to be my book, which I wrote in 2009. And these other references are also good if you want to know more about this. So with this, I end my talk here, and I thank you all for your patient listening. I'll be very happy to answer